Okay, let's wrap up this section in nucleic acids. I already showed you what a nucleotide looks like. Now we're going to be focusing mostly on the nucleic acid portion, right, in the polymer. So just to recap, nucleic acids are formed by joining nucleotides, okay, via dehydration reactions. And I showed you that in my previous video. What do we make? A bunch of covalent bonds between the sugar of one nucleotide and the phosphate of another nucleotide, okay? And we form something called the sugar phosphate backbone, okay? And if you look at this picture over here, okay, actually, there's actually two diagrams here. I want to show you a very simplistic backbone, okay, which is going to be this, okay? It's going to be the yellow and the blue. This is the sugar phosphate backbone, okay? It's made of what? It's made of sugars and phosphates, right? If we pass it over here, that's the same thing. This is going to be our sugar phosphate backbone. As you see here is going to be the prime end is going to be the phosphate end, the 5' end, and then at the bottom you're going to have a 3' end, which is going to be by an OH in the sugar over here. Okay. And how do we add them? We added sugar to our phosphate. That's how we're linking them together, okay, to make the backbone. Okay, so now DNA is going to be double-stranded. It's a double-stranded molecule. Two polynucleotide strands are going to coil around each other, forming something called a double helix. And this is something you probably heard about in terms of DNA. It's in a double helix. Why? Because of its form, because it's double-stranded. The bases, which are these nitrogenous bases, I'm going to have them in a different color. These are the nitrogenous bases, all of the nitrogenous bases. They're going to be linking to each other, okay? The bases they're gonna be forming something called complementary base pairs, okay? And these are going to be linked by hydrogen bonds to bases of the other end or the other strand. So how does that work? Let's look at this picture really quick, okay? Now this is a little more simplified than this picture over here. In this picture over here, what do you see? You see the nitrogenous bases, right, from the different nucleotides. But here it's a little more simplistic, and I want to show you here to show you how everything pairs, okay? This is complementary. This is the complementary base pairs. You have a nucleotide here, okay, connecting to another over here, okay? You want over here to one of the other strand over here, okay? One here to the other over here, okay? And this little dotted line that you see over here, this dotted line, okay, those dotted lines are gonna be hydrogen bonds, okay? And that's how they intertwine, and that's why it makes a double-stranded or double helix type of DNA. That's what makes DNA form a double helix, because of the hydrogen bonds, because of the way one nucleotide is going to be interacting with a nucleotide of another strand. Now, how do we get them together? And this is kind of one of the big points, 3-star concept points, right? How do each nucleotide uh, pair up with another nucleotide? Adenines are always going to be pairing up with thymines, and vice versa. So adenine will always pair with a thymine, or we can say that a thymine can always pair with an adenine. Okay, those are the dance partners. A with T and T with A, adenine with thymine, and thymine with adenine. So far, so good. Copy that. The other dance partners, and this is in terms of DNA. Okay, we'll talk about DNA right now. The other dance partners are going to be guanine, okay, and it's going to be pairing always. It always pairs with cytosine, okay. So we see, and also cytosine always pairs with guanine, okay. That's it. Those are going to be the main pairs, okay. Always. That's the rule. You copy so I can erase. You will never see an adenine connecting with a guanine. No. You will never see an adenine connecting to a cytosine. No, never. Okay? Put it here. Never. Does that make sense? Copy that. You will not see a cytosine connecting to a thymine or a cytosine connecting to a adenine. Never. Does that make sense? I'm not going to continue with the rest, but I just want you to get the idea here. Just want you to get the idea. What's the idea? That the rules are adenine always pairs with thymine, thymine always pairs with adenine. Guanine always pairs with cytosine, cytosine always pairs with guanine. Okay? So there's 4 nitrogenous bases in DNA, as we've been saying, as I already stated. The 4 nitrogenous bases in DNA are going to be adenine, guanine, cytosine, and thymine and cytosine, right? And usually we abbreviate it with adenine with A, G, T, and C. Now, the reason why you have the rule of A with T, T with A, C with G, G with G with C. One of the main reasons why this happens is, okay, it's because adenines, I'm gonna do this in a different color, adenine and guanine, these are gonna be purines, okay? And the purines, as you see over here, the purines are gonna be made of like 2 rings. See, adenine, guanine, they're made of 2 rings, right? Let me do that in another color now. Thymine and cytosine are going to be pyrimidines, different group. And the pyrimidines over here, they're made of 1 ring. Okay, cytosine, thymine, and cytosine. Okay, so remember that. Adenine and guanine, okay, they're both purines. And purines are made of 2 rings. Thymine and cytosine are pyrimidines. And pyrimidines are made of 1 ring. So, and this is going to look a little bit funky, but just bear with me. So why do we always have this rule? A with T, and T with A, C with G, and G with C. Well, I'm going to represent the rings as circles, okay? Just so you get the idea. Adenine is going to be made of 2 rings, right? So I'm just going to simplify this like this. There you go. And thymine is going to be 1 ring, right? You see how that fits? And then let's say I want to have a guanine here that have 2 rings and a cytosine that has 1 ring. You see how they they're going to fit nicely. They're going to be able to connect nicely. What happened? Have an example. Okay, so this is correct. Now, what if I have something like this? I have an adenine with a guanine. Okay, and what would it look like? 2 rings, 2 rings, and then I say have a thymine with a cytosine. 1 ring right here. You see how this doesn't make sense? This doesn't make sense because then this part of the ring is gonna be kind of opening up, right? And this is gonna be too close. There probably won't be no hydrogen bond there. Okay, so that's one of the main reasons why you have this complementary. That's why you always have an A, T, T and A, C and G, and G and C, because you always need to be able to pair, okay, a purine with a pyrimidine. Sorry. And you always have to pair a pyrimidine with a purine. That is why you have to go to the rule. Okay, a purine could be adenine, okay, hanging out with a pyrimidine, cytosine, thymine, for example. Okay, you can have a pyrimidine, for example, pyrimidine would be cytosine. Okay, you can have cytosine, and it's going to be hanging out with a purine, which could be a guanine. Okay. All right. So always remember this rule. Okay, that is extremely important. Remember that rule. Adenine with thymine, thymine with adenine, cytosine with guanine, guanine with cytosine all the time, because a purine has to hang out with a pyrimidine. A pyrimidine has to hang out with a purine always. Now, what determines you? And I'm going to erase this thing really quick. What determines you? The sequence in your DNA. Okay, the sequence of nucleotides. So, for example, I'm going to get you like a gene. I want to say this is going to be a gene. A, T, T, C, C, G. It's supposed to be a G, G, A. That's a gene. That's a sequence. Okay, that sequence is going to determine your proteins. Okay, it's going to determine you, what you look like. Okay, just as an example, again, I'm going to erase this, so please make sure you copy this. You pause this and copy this. I want you to be responsible for this. In class or in the exam, I want to give you a sequence. And you will have to give me the complementary binding. Okay? So the complementary pairing. Apologize. So I'm going to ask you to do something like this. I'm going to give you something like this. It's going to be the sequence. A, T, C, C, G, G, A, T. Right? And I'm going to give you one strand of DNA, which is this one. And you will have give me the other strand, or the complementary strand, of DNA. Okay, so what will be the complementary strand? If this is going to be A, it's going to be bonding with thymine. If we're thymine with adenine, C with guanine, C with guanine, G with cytosine, G with cytosine, adenine with thymine, and thymine with adenine. Okay, I'm going to ask you to do the blue part. Okay, so make sure you know how the pairs go. Make sure you know who the dance partners are here. Okay, and just one more thing, just to stress this point. What is it over here? What are these lines? They represent hydrogen bonds. Okay, which is what it brings them together, and that's why of this structure, because of these hydrogen bonds here. Okay, they're sticking together, and also because these hydrogen bonds between the backbones. Okay, it makes the DNA very stable. Okay, and it makes it go into this double helix kind of shape.